Hello everyone, this is Olivia Crook's honors research on diversity of fungal communities associated with hemlock at Kejimkujik National Park and National Historic Site over the past 30 years. This work is done in collaboration with Dr. Matthew Smith and Dr. Keith Egger, and Olivia is based in Allison Walker's lab at Acadia University Department of Biology. A bit of background on the study, we are capitalizing on some legacy mushroom collections at Acadia in the E.C. Smith Herbarium collected by a master's student, Ivo Polak, who was at Acadia in the 1990s. And Ivo collected from marked plots in Kejimkujik over several years. The specimens were deposited at Acadia, and we're now able to study them and compare them with current mushroom communities in hemlock from the same plots at Keji. We know that hemlocks are a foundational tree species comprising much of the old-growth forest in Nova Scotia, and that Keji is home to some of the largest trees in the province. We know that since 2018, hemlock woolly adelgid has been threatening Keji hemlocks, and is expected to result in 90% decline of hemlock over the next 5 to 15 years. In addition, in 2006, hemlock density in our area was reduced by 50% due to an outbreak of the pale-winged gray moth. Using these legacy and new collections, we're able to look at changes in these important communities over time. We know that fungi have many important and often overlooked roles in forests, and this is true for hemlock stands. These ecological roles include primarily decomposition of dead organic matter, and also degradation and life on wood and woody debris in the forest. We also find ectomycorrhizal fungi associated in symbiotic relationships with hemlock tree roots. So some of the objectives of our study are to compare historic and current fungal communities associated with hemlock in Kejimkujik and identify which fungi are indeed associated with hemlock and thus may be impacted by hemlock decline anticipated to come due to hemlock woolly adelgid. We hypothesize there will be changes in fungal communities, diversity, and abundance compared to the 1990s collections made by Ivo Polak, largely as a result right now of the 2006 pale-winged gray moth infestation, and it may be too soon to detect changes due to hemlock woolly adelgid, but it's important to have this baseline biodiversity data for the park. So our methods include microscopy as well as DNA barcoding to confirm the identifications of the 1990 specimens from the herbarium as well as our new collections. In this way, we can have confidence in our taxonomic identifications and full characterization of these important communities. So we have collected from plot A at Keji this year in August, September, October, and November, and have been drawing mushrooms, extracting DNA, doing PCR, and sending them off for DNA sequencing. So now our task is to compare the current communities with our herbarium specimens, which we have also successfully been identifying using DNA. Preliminary findings of this ongoing study indicate that some of the historic hemlock fungal community is indeed still present at Kejimkujik. And our work continues. We will be providing species lists for the park of fungi present and comparing with older collections from the 1990s. All right, thanks very much.